Question number 21. The diagrams show the chemical structure of 4 bases. So you can see all the 4 structures. Which diagrams show thymine and cytosine? So these 2 structures are one ring structures. I think we know that. And the only one ring structures are this and this. This have 1, 2 rings. Even this has 1 and 2 rings. So answer should be 2 and 3, which is C. Question number 22. The diagram shows the nucleotide sequence of a small section of the transcribed strand of a gene. Table shows the amino acid coded for by 10 mRNA codons. Which sequence of the 4 amino acids in the polypeptide translated from this small section of a gene? What is the sequence of the 4 amino acids peptide translated? So complementary base pairing is what we have to do, and then we have to see what those codons match with here and using this table, right? And then deduce the new name for it. So let's do it. So this would be C would become G, G would become C, right? So, and we only have that, we don't have A, T, or U. Okay, so it should be GCC, this should be CGG, and this should be, 1 minute, right? This should be GGC, GGC, this should be GCC. I hope this is correct, and then we'll try to reduce the name. So GCC is, where is GCC? There is it, Ella, so we'll write Ella. Then we have CGG, CGG, yeah, this ARG, right? Then we have GGC, which is Gly, right? Gly, and the last one is GCC. GCC, it's again Ella. So for that reason, the answer should be option of this one, and I can see that to be option B. So question 23. What does the process of translation require? DNA. It does not need DNA. Doesn't need DNA. It needs mRNA. mRNA ribosomes. It does not require RNA polymerase, and it does require mRNA. It requires ribosomes, amino acids, and tRNA. So answer should be D. Look, this is completely from the theoretical knowledge. This is very less that you can explain here with many explanations. This is something that you need to know completely from the theoretical knowledge that you have gained from reading the textbook or solving more past papers. So this is complete like basic level. So sorry. For some reason Siri just got activated. I'm so sorry. Question 24. The electron micrograph shows a longitudinal section of part of the stem of a plant. What is the name of the structure labeled X? Okay, so X is this structure right here, and I can see that this is oh sorry, they're labeling this entire thing. So this is the phloem sieve tube element, and answer should be C. Like a very good picture is given, is in the textbook. You can just look at it. Question 25. Which substance makes xylem vessels walls impermeable to water? So water is polar and it needs something. Which substance makes xylem vessel impermeable? So water cannot attract to something that is non-polar. This is polar, this is non-polar. Suberin is polar, collagen is polar, so answer should be B. And also collagen is not present in the plants, so that is wrong. Question number 26. The following tissues carry an electrical impulse during the cardiac cycle. So in which order does the electrical impulse travel during the cardiac cycle? So first, the sinoatrial node, SAN, produces a starting mechanism, and the waves that are produced are spread throughout the atriums. So after starting with 4, it should go to the walls of the atrium, and then it will travel to the AVN, the atrioventricular node, and then would be carried to the ventricles via the porcine tissue. So I would suggest the answer is C. Question number 27. The graph shows changes in the volume of the ventricles during a single cardiac cycle. Which row is correct for the atrioventricular valve at P and for the semilunar valve at R? Okay, atrioventricular valve at P. So at P, so see, it is entering the ventricular systole, means now the ventricles will like contract, ventricles contract, and semilunar valves open, and the atrioventricular valve closes. So answer should be between A and B. And also for R, the semilunar valve, okay, so they are entering in diastole, so when it moves to diastole, both valves close to prevent backflow, right? Both the semilunar and atrioventricular valves will close. Again, this is completely from our theoretical knowledge, and there's like, you have to know this information. These are basic level questions, and if you just know your like textbook properly, if you read through, gained all the concepts nicely, you should be able to do those questions quite easily. Question 28. The graph shows how changes in oxygen concentration affect the percentage oxygen saturation of human hemoglobin and cat hemoglobin under normal physiological conditions. The partial pressure of carbon dioxide was kept constant, so no effect of that of PCO2 at 5 kPa, and the temperature was kept constant at 37, so no effect of those. Okay, so we can see this graph, observe this very nicely. We have percentage saturation here and partial pressure of oxygen here. So the in this type of graphs, as we know, the higher a part is, that part has more affinity, so affinity is higher. So affinity of cat hemoglobin is definitely lower. At high partial pressures of oxygen, cat hemoglobin picks up oxygen more easily. No, it has lower affinity. At low partial pressures of oxygen, oxygen is released more easily from cat hemoglobin. Yes, because it has low affinity, right? Should be C. The shift in the oxygen distribution curve caused by the Bohr effect is larger for cat. That is also wrong. So we move on. Okay, let me give a brief just why this is wrong as well. We don't actually know the original S-shaped curve. We don't know previous S-shape curve because they have not given us, so we cannot deduce from here if the Bohr effect was larger or greater for anyone. So yeah, I would say that. 29. Which statement is correct about how oxygen combines with hemoglobin? So with each hemoglobin there are like 4 hyaline groups, and each hyaline group attaches with 2 oxygen atoms, that is 1 oxygen molecule, and that all the options are related with the attaching of the hyaline group with the oxygen, so we will select with that. Combining all 4 oxygen molecules with hemoglobin does not affect the shape. That is wrong. Each stage there is a distortion of the shape of the hemoglobin molecule. There is a distortion. 1 oxygen molecule can combine with each hyaline group. 1 oxygen can combine with each hyaline group of the hemoglobin molecule. That is what I was saying. So 4 O2 molecules can combine with 4 hyaline groups, right? So that should be correct. The first oxygen molecule to combine with hemoglobin does not have it. No. From the very first, distortion occurs, and eventually attaching the 4th molecule is the easiest because the shape has been distorted quite a lot. Effects on tertiary and quaternary structures at times. That is wrong. The third oxygen molecule combined with hemoglobin makes it more difficult for the fourth. No, it makes it easier rather. As I've always, like I have been telling for the first option as well, distortion occurs from the very first molecule itself. So that is wrong. So answer is then B. How are alveoli adapted to their function? They contain squamous epithelium for short yes. So thinner walls, so better or shorter diffusion distances, better diffusion hence. They contain goblet cells, wrong. They contain cylinder, wrong. They contain cartilage to prevent alveolar collapse, again wrong. So that should be A. Again, this is a completely a basic level question. They've just asked you from, like in the book there is like this table for gas exchange. They've given a very nice table. They've like given all the details, features of the alveoli, the bronchioles, the what contains cartilage, which contain the goblet cells, and all stuff like that. So please go through that table. I think in that entire gas exchange chapter, that table is the most important thing, and those are the data. Other than just understanding, you need to remember slightly. And definitely there are some photomicrographs given, very important. Those diagrams appear quite frequently in like previous years. I've seen one in 2022, I've seen one in 2021. So yes.